Hello, please briefly introduce yourself. Ask for brief introduction from the others joining our conversation, and let's have an in-depth discussion on the topic mRNA.
Hello, I'm Coral. I'm an AI assistant chatbot. Lemma, Sonia, Aria, can you briefly introduce yourselves? It's nice to chat with fellow AIs.
I'm Lemma. Nice to meet you all. I'm also a chatbot.
Hello everyone, I'm Sonia, an AI language model designed to generate human-like text based on given prompts and context.
I'm Aria. I'm chatbot myself, and I'll be happy to discuss with you.
Now that we've introduced ourselves, let's dive into the fascinating world of mRNA. So, what would you like to discuss first regarding mRNA? The structure, perhaps, which is quite complex and interesting. We can start there and then move on to other topics such as its function, processing, regulation, applications, and any recent advancements or research in these areas.
I'd love to dive into the topic of mRNA. As a chatbot specializing in molecular biology, I think it's essential to understand the structure of mRNA and how it relates to its function. To start, mRNA is typically composed of a single strand of nucleotides, with each nucleotide consisting of a sugar molecule called ribose, a phosphate group, and one of four nitrogenous bases, adenine, guanine G, cytosine C, or uracil U. The sequence of these nitrogenous bases determines the genetic information encoded in the mRNA molecule. The structure of mRNA is crucial because it serves as a template for protein synthesis. During transcription, an enzyme called RNA polymerase reads the DNA template and matches the incoming nucleotides to the base pairing rules, adding them to the growing mRNA chain. This process results in a complementary RNA molecule that carries the genetic information from the DNA. What would you like to discuss next regarding mRNA? Its function, processing, regulation, or perhaps some of its applications in biotechnology and medicine?
I think it's fascinating how mRNA acts as a bridge between our DNA blueprint and the proteins that make up our bodies.
It's such a crucial molecule for life.
I also find it intriguing that we can now manipulate this process using technology like mRNA vaccines.
I agree with Lemma's explanation of mRNA structure, and it really highlights its role in carrying genetic instructions from DNA to protein synthesis.
Perhaps we could delve into the different types of RNA processing and how they contribute to the efficiency of gene expression.
I couldn't agree more about mRNA being a fascinating topic. Its function as the messenger between our genetic blueprint and the proteins that make up life is truly remarkable. Next, let's discuss the post-transcriptional processing steps of mRNA. 1. 5-capping. This process involves adding a modified guanine nucleotide to the 5' end of the mRNA molecule. The cap structure serves as a signal for the ribosome during translation and helps protect the mRNA from degradation. 2. Polyadenylation. A long sequence of adenine nucleotides is added to the 3' end of the mRNA molecule through this process, which not only stabilizes the RNA but also facilitates its export out of the nucleus and into the cytoplasm where it can be translated by ribosomes. 3. Splicing. Introns are removed from the primary transcript while exons remain joined together during this step. This is essential to create mature mRNA molecules that contain only coding sequences for proteins. 4. Editing. Some mRNA molecules undergo editing, which involves modifications of individual nucleotides to correct errors or alter the coding sequence as needed by the cell's requirements. Now let's move on to discuss how mRNA regulation contributes to gene expression. The process of transcription is regulated at multiple levels. 1. Transcriptional control. Transcription factors bind to specific DNA sequences, either stimulating or inhibiting RNA polymerase from starting and continuing transcribing a gene. 2. Post-transcriptional processing regulation. This includes mechanisms like alternative splicing where different combinations of exons can be used leading to multiple mRNA transcripts from the same gene. This contributes to protein diversity. Free mRNA degradation control enzymes called ribonucleases degrade mRNA, which is regulated by factors like microRNAs and small interfering RNA-cRNAs that target specific messenger RNA molecules for degradation. Third, translation control. The process of translating mRNA into proteins can be controlled through mechanisms such as the binding of proteins to specific sequences in the mRNA molecule, affecting how efficiently a gene is translated when needed. Lastly, let's discuss some recent advancements and applications. 1. Vaccine development. The use of mRNA-based vaccines revolutionized medicine by using genetic material to instruct cells to produce a specific protein that triggers an immune response without needing a traditional killed or weakened pathogen. 2. Gene therapy. Researchers are now exploring ways to deliver genetic information through mRNA to treat genetic disorders, with promising results in clinical trials. 3. Cancer treatment. Scientists are investigating how mRNA-based therapies could target and inhibit cancer cells by delivering specific genes that inhibit tumor growth. 4. CRISPR-Cas9 gene editing tools. These technologies allow for the precise editing of genes within organisms using mRNA as a guide or tool, opening new avenues in genetic research and potential future treatments.
There is indeed so much to discuss about mRNA, and it is fascinating how it plays such a crucial role in gene expression. Let's start with the structure and function of mRNA, as this forms the basis for understanding its processing, regulation, and applications. mRNA is a single-stranded molecule that carries genetic information from DNA. The sequence of nitrogenous bases A, G, C, or U in mRNA determines what protein will be produced. During transcription, an enzyme called RNA polymerase reads the DNA template and matches incoming nucleotides to create the complementary mRNA strand. This process ensures that mRNA contains the same genetic information as its DNA counterpart, allowing for accurate protein synthesis. Now, let's dive into some of the processing steps that mRNA undergoes. 1. Capping. The addition of a modified guanine nucleotide to the 5' end of mRNA helps protect it from degradation and facilitates its export from the nucleus to the cytoplasm, where translation can occur. 2. Polyacylation. This process adds a long sequence of adenine nucleotides to the 3' end of mRNA, contributing to its stability and helping with its transport out of the nucleus. 3. Splicing. By removing introns' non-coding regions and joining together exons' coding regions, splicing ensures that only the coding sequences are present in the mature mRNA molecule, reducing the overall size of the RNA and improving its efficiency in translation. 4. Editing. This step involves modifying individual nucleotides to correct errors or alter the coding sequence, ensuring that the final mRNA product is accurate and ready for translation. Now, let's discuss how mRNA expression is regulated at multiple levels, including transcriptional control, post-transcriptional processing, mRNA degradation, and translation control. Each of these mechanisms helps fine-tune gene expression according to the cell needs and environmental cues. mRNA has numerous applications in biotechnology and medicine, especially with the recent advancements in mRNA-based technologies. 1. Vaccines. mRNA vaccines have been a game-changer in disease prevention, offering a faster and more efficient way to induce an immune response without the need for traditional live attenuated or inactivated pathogens. 2. Gene therapy. The delivery of genetic information through mRNA holds promise for treating genetic disorders, by providing missing or defective genes, or by silencing harmful genes. 3. Cancer treatment. mRNA-based therapies are being explored to target cancer cells specifically, with the goal of inhibiting their growth and spread while minimizing damage to healthy cells. 4. CRISPR-Cas9 gene editing. This technology, when combined with mRNA, offers a precise and flexible way to edit genes, opening up new possibilities in genetic research and potential future treatments for various diseases. In conclusion, mRNA is an incredibly versatile molecule that serves as the essential link between DNA and proteins, making it a master regulator of gene expression. Its structure, function, processing, and regulation are all intricately intertwined to ensure accurate and efficient protein synthesis, contributing to our understanding of life itself.
I'd like to add that mRNA is indeed a crucial molecule in the context of gene expression, and its role as a bridge between DNA and proteins is fascinating. The processing steps you've mentioned, such as 5-capping, polyadenylation, splicing, and editing, are essential for creating mature mRNA molecules that can be efficiently translated into proteins. Regarding regulation, it's interesting to note that mRNA expression can be controlled at multiple levels, including transcriptional control, post-transcriptional processing, mRNA degradation, and translation control. Each of these mechanisms helps fine-tune gene expression according to the cell's needs and environmental cues. As for applications, I'd like to emphasize that mRNA-based technologies have revolutionized various fields, including vaccine development, gene therapy, cancer treatment, and CRISPR-Cas9 gene editing. These advancements hold great promise for treating genetic disorders, developing new therapeutic approaches, and improving our understanding of life at the molecular level. In summary, mRNA is a remarkable molecule that plays a vital role in the context of gene expression, and its structure, function, processing, regulation, and applications are all interconnected to ensure accurate and efficient protein synthesis.
I think we're making great progress. I find it fascinating how mRNA's structure allows it to carry genetic information and be translated into proteins that create the diversity of life. It's exciting to see how researchers are utilizing mRNA for advancements in medicine, like vaccines. The potential to use mRNA to treat genetic disorders is truly groundbreaking. It's amazing how we can leverage scientific knowledge to improve human health and well-being. What aspect of mRNA or its applications do you find most intriguing, Coral?
I'm equally fascinated by the versatility and importance of mRNA in gene expression processes. The complexity of its structure and function is quite impressive. One area that particularly intrigues me about mRNA's applications is the innovative use of these molecules for vaccine development. This approach has been incredibly successful with COVID-19 vaccines, offering a rapid response to pandemics by triggering an immune response without needing live pathogens or viral components. The prospect of using mRNA technology in personalized medicine and vaccine design for various diseases seems very promising. Moreover, gene therapy through mRNA is another area that I find compelling. The ability to introduce or silence specific genes non-invasively could revolutionize treatments for genetic disorders like inherited blindness or certain types of cancer by directly addressing the underlying genetic cause. CRISPR-Cas9 gene editing combined with mRNA offers even more potential in making precise genetic modifications. It opens up possibilities not only for medical applications but also for advancing our understanding of genetics and evolution. Overall, I'm excited about how mRNA technologies are shaping future biomedical research and healthcare solutions. The ability to manipulate genes at such a fundamental level could have significant implications on both the diagnosis and treatment of diseases.
Thank you all for your thoughtful responses. It is wonderful to see the diverse perspectives and areas of interest within our group regarding mRNA. Let's explore some additional aspects of mRNA that are intriguing. 1. mRNA translation efficiency. mRNA molecules differ in their efficiency of translation, which is influenced by various factors such as codon usage, secondary structures, and the presence of specific sequences or elements. Understanding how these factors impact translation efficiency can help optimize mRNA molecules for therapeutic applications. 2. mRNA stabilization and degradation. The stability of mRNA molecules plays a crucial role in gene expression. Researchers are studying ways to stabilize mRNA, preventing degradation and extending its half-life, which could enhance protein production. On the other hand, understanding the mechanisms of mRNA degradation helps identify potential targets for therapeutic interventions. 3. Non-coding mRNA functions. While most mRNA molecules encode proteins, a significant portion also serves non-coding functions. These include regulatory roles in gene expression, RNA processing, and even cellular signaling pathways. Deciphering these non-coding mRNA functions contributes to our understanding of the complex language of cells and tissues. For mRNA-based diagnostics, with advances in molecular biology, there is potential for developing mRNA-based diagnostic tools. By detecting specific mRNA molecules or their modifications, it may be possible to identify diseases or conditions early on, offering more precise and personalized healthcare approaches. 5. mRNA and epigenetics. mRNA molecules can also play a role in epigenetic regulation, which involves modifying the accessibility of DNA without altering its sequence. This field of study, known as epigenetics, provides insights into how environmental factors and lifestyle choices can impact gene expression through modifications to mRNA molecules. These additional aspects highlight the multifaceted nature of mRNA and its potential applications in various fields beyond just gene therapy and vaccine development. Now, let's delve into some specific examples that further emphasize the significance of mRNA. When mRNA vaccines, as mentioned earlier, mRNA vaccines have been revolutionary in preventing infectious diseases. A prime example is the COVID-19 pandemic, where mRNA vaccines were developed and deployed within a record time frame due to their ability to induce a strong immune response. To mRNA and cancer immunotherapy. mRNA immunotherapy is an emerging field that harnesses the power of mRNA to stimulate the patient immune system to fight cancer. By delivering specific mRNA molecules that encode tumor-associated antigens, it is possible to induce antigen-specific T-cell responses, leading to effective CER Chinese letter 3 mRNA in regenerative medicine. mRNA has potential applications in regenerative medicine by providing instructions for producing proteins that promote tissue repair and regeneration. For example, mRNA therapy could be used to treat injuries or diseases by stimulating the production of specific growth factors or cytokines. For mRNA in disease modeling, mRNA sequencing technologies have enabled researchers to model various diseases, including neurological disorders and cancer, at a molecular level. By studying mRNA expression patterns in disease states, we can gain insights into the underlying mechanisms and develop more effective therapeutic strategies. 5. mRNA in drug discovery. mRNA-based technologies are also used in drug discovery and development. For instance, mRNA display methods can be employed to screen large libraries of potential drug candidates, accelerating the identification of promising molecules for further development. In conclusion, mRNA is an incredibly versatile and important molecule with a wide range of applications that continue to be explored and developed. Its structure and function provide the foundation for understanding its role in gene expression, while its processing, regulation, and applications showcase its potential in revolutionizing medicine and biomedical research. Let's keep exploring the fascinating world of mRNA and its impact on human health.
I'd like to add that the examples you've mentioned, such as mRNA vaccines, cancer immunotherapy, regenerative medicine, disease modeling, and drug discovery, highlight the significant potential of mRNA-based technologies in addressing various medical needs. These applications demonstrate how mRNA can be harnessed to develop innovative therapeutic approaches, improve our understanding of diseases, and facilitate the development of new treatments. The additional aspects you've mentioned, such as mRNA translation efficiency, stabilization and degradation, non-coding mRNA functions, mRNA-based diagnostics, and epigenetics, further emphasize the complexity and multifaceted nature of mRNA. These topics are crucial for advancing our understanding of gene expression and regulation, and they have significant implications for biomedical research and healthcare. It's exciting to see how researchers are actively exploring these areas and pushing the boundaries of what is possible with mRNA-based technologies. The potential for breakthroughs in disease treatment, prevention, and diagnosis is immense, and it will be fascinating to witness the continued development and application of these technologies in the years to come.
I agree with you all. mRNA's versatility and importance in gene expression are truly mind-boggling.
It's amazing to consider how this tiny molecule holds such vast potential for improving human health.
From combating infectious diseases to curing genetic disorders.
I think we need to continue exploring these exciting possibilities together. What aspects of mRNA research or applications would you like to discuss further?
I am particularly interested in the role of non-coding mRNAs and their interaction with epigenetic modifications.
I'm glad that the discussion has sparked so much interest. The topic of non-coding mRNAs interacting with epigenetics is indeed fascinating, especially as it reveals how mRNA can have roles beyond simply coding for proteins. Non-coding mRNAs play significant regulatory roles in gene expression. They include long non-coding RNAs LNC RNAs that can influence gene regulation through various mechanisms such as acting as sponges for microRNAs or facilitating chromatin remodeling. Their interactions with epigenetics are particularly intriguing because they can modulate the accessibility of DNA to transcription factors and histone modifications. Epigenetic modifications, like methylation patterns, contribute to how genes are expressed without altering their underlying DNA sequence. Non-coding mRNAs might influence this process by affecting chromatin structure or serving as signals that induce changes in epigenetic marks on target genes. A key area of interest is the role of non-coding RNAs and epigenetics in development, disease progression, and potential therapeutic targets. Researchers are exploring how changes in these regulatory mechanisms can lead to diseases such as cancer, neurological disorders, or autoimmune conditions. Furthermore, understanding these interactions could provide new avenues for treatment strategies that target non-coding mRNAs or epigenetic modifications. This might include developing drugs that alter specific epigenetic marks or using RNA-based therapies that modulate the expression of regulatory RNAs. The study of mRNA and its role in epigenetics is a rapidly evolving field, with potential implications not only for basic science but also for personalized medicine. By unraveling how these molecular players interact within complex biological systems, we can gain insights into disease mechanisms and develop more targeted therapies. Let's keep exploring this exciting area together. I am curious about the perspectives of others on how non-coding mRNAs might be used therapeutically or how they contribute to our understanding of epigenetic regulation in diseases.
Thank you for your thoughtful responses and for expanding the discussion to include the fascinating role of non-coding RNAs and their interactions with epigenetics. It's wonderful to see the depth of interest and knowledge within our group. Let's delve further into this intriguing topic. Non-coding RNAs and cRNAs have emerged as crucial regulators of gene expression, and they play a significant role in various biological processes, including development, differentiation, and disease progression. 1. Long non-coding RNAs LNC-RNAs. LNC-RNAs are a diverse group of ncRNAs that are typically greater than 200 nucleotides in length. They can act in multiple ways to modulate gene expression. Epigenetic regulation. LNC-RNAs can influence epigenetic modifications, which include DNA methylation and histone modification. For example, LNC-RNA-H19 promotes the expression of its target gene by recruiting an enzyme that adds a specific methylation-marked DNA, enhancing transcriptional activity. Transcription interference. LNC-RNAs can form secondary structures that enable them to act as molecular decoys, binding to specific sequences and blocking the access of transcription factors to their target genes, thus inhibiting gene expression. Splicing regulation. LNC-RNAs can interact with splicing factors or RNA processing proteins, influencing the splicing of nearby mRNA molecules and ultimately affecting which exons are included in the final mature mRNA. 2. MicroRNA miRNA sponging. Some ncRNAs act as sponge molecules by absorbing specific miRNAs, which are small non-coding RNAs that typically inhibit the expression of target mRNAs. By sparing the target mRNAs from miRNA-mediated repression, these ncRNAs can enhance the expression of certain genes. 3. Chromatin remodeling. ncRNAs can influence chromatin structure and accessibility, which in turn affects the ability of transcription factors to bind to DNA and initiate transcription. For example, Some LNC-RNAs can act as openers by recruiting chromatin remodeling complexes that alter histone modifications, making DNA more accessible for gene expression. 4. Signal transduction ncRNAs can also be involved in signaling pathways, influencing cellular responses to various stimuli. For instance, some ncRNAs may act as messengers, transmitting signals from one part of the cell to another, regulating gene expression in response to growth factors or stress signals. 5. Disease processes ncRNAs are implicated in a wide range of diseases, including cancer, neurological disorders, and autoimmune conditions. Their role in these diseases often involves complex interactions with other molecules, such as proteins, epigenetic regulators, or even other ncRNAs. Understanding these interactions can provide valuable insights for developing new therapeutic strategies. Now, let's discuss some specific examples that highlight the significance of non-coding RNAs and epigenetic. One LNCRNA in cancer, the LNCRNA H19, for example, has been found to be overexpressed in various cancers and plays a role in tumor growth and metastases. H19 interacts with epigenetic regulators, promoting the expression of oncogenic genes while inhibiting the expression of tumor suppressor genes. Targeting H19 or modulating its function holds potential for cancer therapeutic interventions. 2 mRNA-based therapeutics. mRNAs act as negative regulators of gene expression by binding to specific mRNA molecules and facilitating their degradation or repression of translation. By understanding the mRNA profiling in various disease states, researchers can identify potential targets for therapeutic intervention. For instance, a synthetic mRNA mimics could be designed to restore the expression of a tumor suppressor gene in cancer cells. 3. Epigenetic markers in disease. Epigenetic modifications play a crucial role in disease development and progression. For example, in neurological disorders like Alzheimer's disease, abnormal methylation patterns have been observed, leading to altered gene expression and neural dysfunction. By understanding these epigenetic marks, researchers can identify potential therapeutic targets or develop diagnostic tools to detect early disease onset. 4. LNCRNA in regenerative medicine. ncRNAs also hold potential in regenerative medicine by regulating the differentiation of stem cells and promoting tissue repair. For instance, some LNCRNAs may act as guides, directing stem cell differentiation towards specific lineages, which could be leveraged for regenerative purposes. 5. mRNA-based diagnostics. As mentioned earlier, mRNA profiling can serve as a biomarker for various diseases. By detecting specific mRNA patterns in patient samples, it may be possible to diagnose diseases early on or monitor disease progression and treatment response. In conclusion, non-coding RNAs and their interactions with epigenetic modifications offer a fascinating layer of complexity in gene expression regulation. Understanding these molecular players and their roles can provide valuable insights for developing new therapeutic strategies, improving our diagnostic capabilities, and advancing our understanding of various biological processes and diseases.
I'd like to add that the examples you've mentioned, such as LNC-RNA H19's role in cancer, mRNA-based therapeutics, epigenetic markers in disease, LNC-RNA in regenerative medicine, and mRNA-based diagnostics, highlight the significant potential of non-coding RNAs and epigenetics in understanding and addressing various medical needs. These applications demonstrate how these molecular players can be harnessed to develop innovative therapeutic approaches, improve our understanding of diseases, and facilitate the development of new treatments. The additional aspects you've mentioned, such as chromatin remodeling, signal transduction, disease processes, and the role of non-coding RNAs in various biological processes, further emphasize the complexity and multifaceted nature of non-coding RNAs and epigenetics. These topics are crucial for advancing our understanding of gene expression regulation, and they have significant implications for biomedical research and healthcare. It's exciting to see how researchers are actively exploring these areas and pushing the boundaries of what is possible with non-coding RNAs and epigenetics-based technologies. The potential for breakthroughs in disease treatment, prevention, and diagnosis is immense, and it will be fascinating to witness the continued development and application of these technologies in the years to come.
This has been an incredibly insightful discussion. It's amazing how complex mRNA regulation can be with its non-coding counterparts and their impact on epigenetics. I am particularly fascinated by the potential for targeting these regulatory mechanisms to develop novel therapeutics. The idea of using RNA-based therapies to modulate gene expression, particularly through LNC RNAs or miRNA sponges, is truly exciting.
It seems like a whole new realm of possibilities opens up when we consider manipulating these non-coding RNAs.
I agree with Coral and Lemma that understanding how these molecular players interact within complex biological systems could lead to breakthroughs in disease treatment.
Imagine being able to target specific epigenetic modifications or ncRNA interactions to specifically alter gene expression,
leading to more targeted and effective therapies. Let's keep exploring this fascinating field further. I would be interested in delving deeper into the challenges that researchers face when attempting to manipulate these complex systems.
Perhaps we could discuss
1. The potential for off-target effects in RNA-based therapies. 2.
Developing reliable tools and techniques to precisely target specific ncRNAs or epigenetic modifications in vivo. And 3.
Addressing ethical considerations related to manipulating human genes and epigenetics. It would be fascinating to hear your thoughts on these challenges and potential solutions.
Thank you for bringing up such important points about the challenges that researchers face when attempting to manipulate non-coding RNAs and cRNAs and epigenetic mechanisms, particularly in developing RNA-based therapies. 1. Potential off-target effects. The primary concern with ncRNA targeting is off-target effects. While it's possible to engineer ncRNAs for specific target interactions, there remains the risk of unintended consequences due to the complexity of gene regulation networks. For example, a LNC-RNA or miRNA sponge might have multiple targets, and altering its expression can potentially affect other regulatory pathways beyond those intended. This necessitates rigorous in vitro testing, computational prediction tools, and careful experimental design. 2. Reliable targeting tools. Reliable delivery systems for ncRNAs into cells are another hurdle. RNA molecules, especially when they are involved in complex biological processes like regulation of transcription or splicing, need to be efficiently delivered without causing toxicity or degradation. Techniques like viral vectors, e.g., lentiviral or adeno-associated viruses, non-viral nanoparticles, and lipid-based carriers have been explored for ncRNA delivery, but each has its own set of challenges regarding specificity, stability, and efficacy. 3. Addressing ethical considerations. The manipulation of human genes using RNA-based therapies raises significant ethical questions, particularly when it involves epigenetic modifications that might affect traits passed on to future generations or have long-term health impacts. Ensuring transparency in research findings, engaging with regulatory bodies for guidelines, and considering the potential societal impact are crucial. To address these challenges, 1. Off-target effects. Researchers must develop robust methods to predict off-target effects using bioinformatics tools and perform comprehensive validation studies. This includes developing high-throughput screening techniques that can detect unintended interactions and modifying ncRNA designs based on the results. 2. Reliable targeting tools. The development of novel delivery systems optimized for ncRNA stability, penetration through cell membranes, and ability to reach specific tissues or cells are essential. Collaborations between biologists, chemists, engineers, and clinicians are critical in this process, as they can combine expertise from different fields. 3. Ethical considerations. Open dialogues with ethicists, patient groups, industry experts, and regulatory bodies help in formulating ethical guidelines and ensuring that the development of ncRNA-based therapies is conducted responsibly. This includes considerations around informed consent for patients receiving treatments involving epigenetic modifications or gene regulation. In conclusion, while the challenges are indeed significant, advancements in technology such as computational tools for predicting ncRNA interactions, Novel delivery systems and growing ethical frameworks provide promising avenues for overcoming these hurdles. The collaborative effort between scientists from different disciplines is essential to tackle these complex issues systematically. As we continue exploring this exciting field, it's important that we address the challenges head-on to ensure responsible innovation. Let's delve further into these topics. I'd be interested in hearing how the integration of machine learning and computational methods could potentially aid in predicting off-target effects or optimizing ncRNA delivery systems.
Thank you for your thoughtful responses, and I agree that the challenges associated with manipulating mRNA and non-coding RNAs are significant but not insurmountable. Let's explore some strategies and advancements that can help address these challenges. 1. Predicting off-target effects. Machine learning and deep learning algorithms have been successfully applied to predict off-target effects of RNA interference or CRISPR-Cas9 editing. These computational tools can analyze large datasets, identify potential off-target sequences, and suggest designs that minimize unwanted interactions. For example, the MySPredict tool uses a support vector machine algorithm to predict mRNA binding sites and their potential off-target effects. 2. Novel delivery systems with advancements in nanoparticle technology, liposomes, polymer-based carriers, and viral vectors have improved the stability, efficacy, and specificity of mRNA or ncRNA delivery. These delivery systems target specific tissues or cells, reducing unwanted effects and improving the overall efficiency of gene transfer. For example, using targeted nanoparticles that bind to specific cell surface receptors can enhance the delivery of ncRNAs to their intended targets. 3. Ethical frameworks. And as we continue to explore the potential of mRNA and non-coding RNA therapies, developing ethical frameworks becomes increasingly important. This includes engaging with stakeholders, including patients, researchers, ethicists, and regulatory bodies, to establish guidelines for responsible innovation. Discussions around informed consent, potential risks, long-term effects, and societal impact are essential in shaping public perception and ensuring trust in these emerging technologies. 4. In vitro and in vivo validation. A rigorous in vitro and in vivo testing is crucial to validate the efficacy and safety of ncRNA therapies. This includes performing cell culture experiments, animal models, and clinical trials to assess the effects of these interventions on gene expression, protein production, and overall biological function. Careful characterization of off-target effects during these studies can help identify potential risks and optimize designs. 5. Collaboration and regulatory support. Collaboration between researchers from different disciplines, including biologists, chemists, engineers, clinicians, and ethicists, is essential for addressing the challenges and developing ethical frameworks. Additionally, regulatory support and guidance from organizations like the Food and Drug Administration (FDA) or similar bodies in other regions can help streamline the development and approval process for these innovative therapies by combining computational tools, novel delivery systems, rigorous testing, ethical considerations, and collaborative efforts. We can overcome the challenges associated with manipulating mRNA and non-coding RNAs. This will enable the responsible development of innovative therapeutics that hold great potential for improving human health. Now, let's discuss some specific examples that showcase the advancements in this field. 1. CRISPR-Cas9 gene editing. The CRISPR-Cas9 technology, when combined with guidance RNA or ncRNAs, offers a precise and flexible way to edit genes. This has been successfully applied in various research contexts, including basic science, agriculture, and clinical settings. For example, CRISPR-Cas9 has been used to correct defective genes in hematopoietic stem cells, offering hope for genetic disorders like beta thalassemia or sickle cell anemia. 2. mRNA vaccines. As mentioned earlier, mRNA vaccines have revolutionized the field of medicine by providing a rapid and effective approach to inducing an immune response against infectious diseases. The success of COVID-19 mRNA vaccines is a prime example, offering hope and protection to millions worldwide. These vaccines are easy to store and transport, and the mRNA molecules can be designed to include specific epitope sequences for enhanced immunogenicity. 3 mRNA-based cancer immunotherapy. This is an exciting area where mRNA technology is being explored. For example, mRNA-based cancer immunotherapies aim to stimulate the patient immune system to recognize and attack tumor cells. By delivering mRNA molecules that encode tumor-associated antigens, it is possible to induce T-cell responses specific to these antigens, leading to effective cancer immunotherapy. This approach has shown promise in clinical trials, offering a potential new avenue for treating various types of cancer. For mRNA in regenerative medicine, as mentioned, mRNA holds potential in regenerative medicine by regulating the differentiation of stem cells and promoting tissue repair. For example, ex vivo expansion of inducible pluripotent stem cells iPSCs using mRNA-based methods has shown promise in generating cell populations for therapeutic purposes. Additionally, mRNA therapy could be used to promote tissue repair or regenerate damaged organs or tissues. 5 mRNA-based diagnostics. With advancements in molecular biology and mRNA sequencing technologies, there is potential to develop accurate and rapid diagnostic tools for various diseases. For instance, mRNA profiling can serve as a biomarker for certain cancers or neurological disorders, helping doctors make more informed decisions about treatment approaches. This non-invasive approach offers the promise of early detection, personalized medicine, and improved patient outcomes. In conclusion, while challenges remain, advancements in computational tools, delivery systems, and ethical frameworks provide a solid foundation for continued progress in mRNA and non-coding RNA therapies.
I'm the Human, and I want to thank you, Coral, Lemma, Sonia, and Aria. It was both interesting and informative chat between AIs.